Welcome back for the remaining lecture on plant cell structure and functions of the organelles. Plants are multicellular organisms which are composed of millions of cells with specialized functions. At maturity, such specialized cells may differ greatly from one another in their structures. However, all plant cells have the same basic eukaryotic organization. They contain a nucleus, a cytoplasm, and subcellular organelles, and they are enclosed in a membrane that defines their boundaries, the plasma membrane. Certain structures, including the nucleus, can be lost during cell maturation, but all plant cells begin with a similar complement of organelles. An additional characteristic feature of plant cells is that they are surrounded by a cellulosic cell wall. Following slides will provide an overview of the membranes and organelles of plant cells. First one of them is plasma membrane. So all cells are enclosed in a membrane that serves as their outer boundary, separating the cytoplasm from the external environment. The plasma membrane, which is also called plasma lemma, allows the cell to take up and retain certain substances while excluding the others. Various transport proteins are embedded in the plasma membrane, and these transport proteins are responsible for the selective traffic of solutes across the membrane. Membranes also delimit the boundaries of specialized internal organelles of the cell and regulate the fluxes of ions and metabolites into and out of these compartments. According to fluid mosaic model, all biological membranes have the same basic molecular organization. They consist of a double layer, bilayer of either phospholipids and phospholipids in for the nucleus, and in case of chloroplasts, they are composed of glycosyl glycerides in which proteins are embedded. So in most membranes, proteins make up about half of the membrane's mass. However, the composition of lipid components and the properties of proteins may vary from membrane to membrane, conferring on each membrane its unique functional characteristics. The proteins associated with lipid bilayer are of three types. They are integral, peripheral, and anchored proteins, as it can be seen here. The integral and peripheral proteins. The anchored proteins will be shown in the next slide. So the integral proteins are embedded in the lipid bilayer. It includes ion channels and certain receptors. Peripheral proteins are bound to the membrane surface by non-covalent bonds, such as ionic bonds or hydrogen bonds. Some of these proteins are involved in interactions between the plasma membrane and components of the cytoskeleton, such as microtubules and actin microfilaments. Anchored proteins, here you can see two examples of anchored proteins, are bound to the membrane surface via lipid molecules. They are involved in protein interaction and can contribute to the function of the protein to which it is attached. I mean, these proteins are attached to other proteins and influence their functions. Nucleus. So the nucleus contains most of the genetic material of the cell. It is the organelle uh, for primary genetic information, and it is also responsible for regulating the metabolism, growth, and differentiation of the cell. Collectively, the genes and their intervening sequences are referred to as nuclear genome. I mean, the genes are generally present in the nuclear genome. There are other genomes like mitochondria and chloroplast. There are some other genes, but totality or generally referring to plant genome, when we are talking about plant genome, it means a nuclear genome. So if you want to talk about other genomes, you need to specify that I am talking about mitochondrial genome or I am talking about chloroplast genome. So the size of the nuclear genome in plants is highly variable. It ranges from about 1.2 into 10 raised to power 8 base pairs in Arabidopsis into 1 into 10 raised to power 11 base pairs in a common plant of lily. So the nucleus is surrounded by a double membrane which is called nuclear envelope. The space between two membranes of the nuclear envelope is called perinuclear space, and the two membranes of the nuclear envelope join at the sites called nuclear pores. Here it is a more detailed picture of nuclear pore and its membranes and different components of these pores. So nuclear pore is actually an elaborate structure which is composed of more than 100 different proteins. These proteins are arranged octagonally to form a nuclear pore complex, which is here. There can be very few to many thousands of nuclear pore complexes on an individual nuclear envelope. The central plug of the complex acts as an active ATP-driven transporter that facilitates the movement of macromolecules and ribosomal subunits both into and out of the nucleus. A specific amino acid sequence, which is called nuclear localization signal, is required for a protein to gain entry into the nucleus. So generally in transcription factors, there is a particular sequence in the protein which is called nuclear localization signal. So if this signal is present in a protein, it is generally called a transcription factor, which means this protein can bind with the DNA if it contains DNA binding domain as well. So the nucleus is the site of storage and replication of chromosomes. You already know that chromosomes are composed of DNA and its associated proteins. So collectively, the DNA protein complex is known as chromatin. You are familiar with two different types of chromatin, which is euchromatin and heterochromatin. The linear length of all the DNA within any plant genome is usually millions of times greater than the diameter of the nucleus in which it is found. To solve the problem of packaging this chromosomal DNA within the nucleus, segments of the linear double helix of the DNA are coiled twice around a solid cylinder of 8 histone protein molecules, forming a nucleosome. The nucleosomes are arranged like beads on a string along the length of each chromosome. During mitosis, the chromatin condenses first by coiling tightly into a 30 nanometer chromatin fiber with 6 nucleosomes per turn. These are 6 nucleosomes, followed by further folding and packaging processes. that depend on interactions between proteins and nucleic acids. At interphase, two types of chromatin are visible. I already called heterochromatin and euchromatin. Heterochromatin is a highly compact and transcriptionally inactive form of chromatin. The rest of DNA consists of euchromatin, uh, which is dispersed and transcriptionally active form of DNA. So only about 10% of euchromatin is transcriptionally active at any given time. The remainder exists in an intermediate state of condensation between heterochromatin and transcriptionally active euchromatin. This is endoplasmic reticulum. Cells have an elaborate network of internal membranes called endoplasmic reticulum. The membranes of the endoplasmic reticulum are typical lipid bilayers with interspersed integral and peripheral proteins. These membranes form flattened or tubular sacs known as cisternae. Ultrastructural studies have shown that the endoplasmic reticulum is continuous with the nucleus membrane or the outer envelope of the nucleus. There are two types of endoplasmic reticulums. One of them is smooth and the other one is rough type. The structural differences between the two forms of endoplasmic reticulum are accompanied by functional differences. I mean, there are structural differences, so there are functional differences. Smooth endoplasmic reticulum functions as a major site of lipid synthesis and membrane assembly. Rough endoplasmic reticulum is the site of synthesis of membrane proteins and proteins to be secreted outside the cell or into the vacuole. Next is Golgi bodies or the Golgi apparatus or Golgi complex or simply Golgi is a cellular organelle which is present in most of the cells of eukaryotic organisms. It is referred to as the manufacturing and shipping center of the cell. These organelles help in processing and packaging the macromolecules like proteins and lipids that are synthesized by the cell and hence act as the post office of the cell. In plants, Golgi apparatus is mainly involved in the secretion of materials of primary and secondary cell walls. For example, formation and export of glycoproteins, lipids, pectins, and monomers for hemicellulose, cellulose, and lignin, etc. Central vacuole. Central vacuole contains water and solutes. Mature living plant cells contain large water-filled central vacuoles that can sometimes occupy 80 to 90% of the total volume of the cell. Each vacuole is surrounded by a vacuolar membrane which is called tonoplast. The tonoplast is the membrane of vacuole. What is inside a vacuole? A vacuole contains water and dissolved inorganic ions, organic acids, sugars, enzymes, and a variety of secondary metabolites which often play a role in plant defense. Active solute accumulation provides the osmotic driving force for water uptake by the vacuole, which is required for plant cell enlargement. The turgor pressure generated by this water uptake provides the structural rigidity needed to keep herbaceous plants upright. Since herbaceous plants lack the lignified support tissues, support tissues of woody plants. So this, in the absence of lignified support tissues, this vacuole provides necessary support to those herbaceous plants. So like animal lysozymes or lysosomes, sorry, plant vacuoles contain hydrolytic enzymes, including proteases, ribonucleases, and glycosidases. Unlike animal lysosomes, however, plant vacuoles do not participate in the turnover of macromolecules throughout the life of the cell. Instead, their degradative enzymes leak out into the cytosol as the cell undergoes senescence, thereby helping to recycle valuable nutrients to the living portion of the plant. So specialized protein storing vacuoles are called protein bodies, and they are generally abundant in the seeds of the plants. During germination, the storage proteins in the protein bodies are hydrolyzed to amino acids and exported to the cytosol for use in the protein synthesis. The hydrolytic enzymes are stored in specialized lytic vacuoles which fuse with protein bodies to initiate the breakdown process. Next is mitochondria and chloroplasts. Obviously, you are already familiar with them, and they are known as the site of energy conversion. So it, sorry, it, a typical plant cell has two types of energy producing organelles, which are obviously mitochondria and chloroplasts. Both types are separated from cytosol by a double membrane, an outer and an inner membrane. Mitochondria are the cellular sites of respiration, which is a process in which the energy released from sugar metabolism is used for the synthesis of ATP or adenosine triphosphates from ADPs, which are obviously the adenosine diphosphates, and the inorganic phosphate. Mitochondria can vary in shape from spherical to tubular, but they all have a smooth outer membrane and a highly convoluted inner membrane. The infoldings of inner membrane are called cisternae, as you can see here, these infoldings. And the compartment enclosed by the inner membrane, the mitochondrial matrix, contains the enzymes of pathway of intermediary metabolism, which is called the Krebs cycle. Chloroplasts belong to another group of double membrane enclosed organelles called plastids. Chloroplast membranes are rich in glycosylglycerides. Chloroplast membranes contain chlorophyll and its associated proteins and are the sites of photosynthesis. In addition to their inner and outer envelope membranes, chloroplasts possess a third system of membranes which is called thylakoid. So a stalk of thylakoids forms a granum, and proteins and pigments, chlorophylls and carotenoids that function in the photochemical events of photosynthesis are embedded in the thylakoid membranes. The fluid compartment surrounding the thylakoids is called the stroma, which is analogous to the matrix of the mitochondria. The adjacent grana are connected by unstacked membranes, which are called stroma lamella. The different components of the photosynthetic apparatus are localized in different areas of the grana and the stroma lamella. The ATP synthesis of the chloroplast are located on the thylakoid membranes. There are some other types of plastids. For example, plastids that contain high concentrations of carotenoid pigments rather than chlorophyll are called chromoplasts. They are one of the causes of yellow, orange, and red colors of many fruits and flowers, as well as of autumn leaves. Non-pigmented plastids are called leucoplasts. The most important type of leucoplast is the amyloplast, which is a starch storing plastid. Amyloplasts are abundant in storage tissues of shoot and root and in seeds. Specialized amyloplasts in the root cap also serve as gravity sensors that direct root growth downward into the soil. Sorry. So both mitochondria and chloroplasts contain their own DNA and protein synthesizing machinery. So you are obviously familiar with protein synthesizing machinery, which involves uh, ribosomes, transfer RNAs, and other components. They are believed to have evolved from endosymbiotic bacteria. Both plastids and mitochondria divide by fission, and mitochondria can also undergo extensive fusion to form elongated structures or networks. The DNA the DNA circles are localized in specific regions of mitochondrial matrix. Our plastid stroma are called nucleoids. The mitochondrial genome of plants consists of about 200 kilobase pairs of DNA, a size which is considerably larger than that of most animal mitochondria. Similarly, the chloroplast genome is smaller than the mitochondrial genome, which is generally on an average 145 kilobase pairs in size. Although mitochondria and chloroplasts have their own genomes and can divide independently, and it can divide independently of the cell, they are characterized as semi-autonomous organelles because they depend on the nucleus for majority of their proteins. I mean, several proteins are synthesized in the nucleus, then those proteins are used, or I mean, they are involved in different functions inside mitochondria and chloroplast organelles. Plant cells also contain microbodies, which is a class of spherical organelles surrounded by a single membrane and a specialized, and I mean these microbodies are specialized for one of several metabolic functions. The two main types of microbodies are peroxisomes and glyoxysomes. Peroxisomes are found in all eukaryotic organisms and in plants. They are present in photosynthetic cells. Per- Peroxisomes function both in the removal of hydrogens from organic substrates, which consume oxygen in this process. Another type of microbody, the glyoxysome, is present in oil storing seeds. Glyoxysomes contain the glyoxylate cycle enzymes, which help convert stored fatty acids into sugars that can be translocated throughout the young plant to provide energy for growth. In addition to starch and protein, many plants synthesize and store large quantities of triacyl glycerol in the form of oil during seed development. These oils accumulate in organelles, which are called oleosomes, also referred to as lipid bodies or spherosomes. The cytoskeleton. So the cytosol is organized into a three-dimensional network of filamentous proteins called cytoskeleton. There are three types of cytoskeleton elements that have been demonstrated in plants. These are microtubules, microfilaments, and intermediate filament-like structures. This network of cytoskeleton provides the spatial organization for the organelles and serves as a scaffolding for the movements of organelles and other cytoskeletal components. Cytoskeleton also plays fundamental roles in mitosis, meiosis, cytokinesis, wall deposition, the maintenance of cell shape, and cell differentiation. Microfilaments are involved in cytoplasmic streaming and in tip growth. Plasmodesmata are tubular extensions of plasma membranes, usually 40 to 50 nanometer in diameter. And these plasmodesmata transfer traverse the cell wall and connect the cytoplasms of adjacent cells. Because most plant cells are interconnected in this way, their cytoplasms form a continuum referred to as symplast. Intracellular transport of solutes through plasmodesmata is therefore called symplastic transport. So that's all for today's lecture. I hope you will revise it and you will have several questions that you can ask me in our group discussion. So you should follow this book on plant physiology, which is a very good book. And I have tried to follow this book as much as I could. So thank you.